Um, Good morning, good afternoon, good evening. Hello for everyone. I'm very pleased to introduce you to our mentor, Veronica Shubayev. We invite you to an exciting journey into the world of space. Let's get started. Veronica, please.
Hi, everyone. Thank you for tuning in. Today, I'm going to be talking about how to conduct biological research from idea to discovery. Thank you, Triple V. Thank you, Elena, for the important work that you do. And thank you for this opportunity to contribute. So today, I'm going to talk about the questions that you posed, how research is born, experimental design, analysis, and troubleshooting. We're going to talk about some of the examples of youth research, how to start your own project, and we can talk questions and answers if you have it. So, how is research born? I think this stereotype of the idea coming to your head is this eureka moment. And that happens very commonly when we think about our research often. When you just start your process, you can start with simple questions. I think the important thing is to have the questions that have big impact. A question that interested me was why and how we feel pain. And before I tell you why I think this is a question of big impact, I would like to invite you to pause and think and write down which unsolved problems of the society you resonate with, touch you very deeply, and which of you like to solve or be a part of solving if it's a very big problem. So write it down. We'll come back to that. So, chronic pain is a major issue. Um, it's a complex issue. It's a biological, it's an emotional, it's a philosophical issue that interested me to parse it out. It's debilitating. It's number one reason why patients come to doctor's office. Worldwide, 1.5 billion of people suffer from chronic pain. In the US alone, it's over $650 billion a year is the cost of medical expenses and um, lost productivity. And of course, a major issue is opioid crisis. But this is not a new question. Of course, this is from the antiquity. Aristotle enumerated five senses. He didn't name pain among them. He thought pain was an emotional issue. He called it passion of the soul. Then Hippocrates did think of pain as a biological issue, but he thought of a heart as a pain organ, and that the fluids, vital fluid imbalance is the cause of it. Then Avicenna was the one who actually enumerated pain as a sense, and only in 1600s by René Descartes, who did a phenomenal job in characterizing numerous medical and biological problems. He was the first to characterize the pathway of pain where fire fibrils would enter the body and signal to the brain, and then from the brain back to the foot. And then we would remove the foot through the muscle contraction. Almost 400 years later, we know a lot of things about mechanisms of pain. We know different types of pain. We know chemical, biological, structural, genetic, epigenetic mechanisms of pain. We also know that expectation and mood and prior experience and thinking of pain catastrophizing is going to affect it. That's why modalities like meditation and education are important in managing pain. So what what is pain? And um, going along with the idea of making the biggest impact, what is the worst type of pain? We need pain. Pain is an evolutionary alarm from the impending danger. Um, so there is good pain. And then there is pain when tissue is injured, inflammatory pain, and it lasts for as long as the tissue injury lasts. And then there is this pain which doesn't go away even after injury is recovered. It can be spontaneous. It gives these shooting, burning, pins and needles, stabbing, electrical shock-like sensations. This is nerve injury pain inside those organs and or called neuropathic pain. So it happens after nerve injury by trauma or tumor, back pain, drugs and toxins like alcohol, chemotherapy, different viruses can affect the nerves and cause neuropathic pain. About 30% of pain is due to diabetes. There's also immune and idiopathic. Idiopathic when we don't know the cause. Basically, a patient comes and doesn't know why. This is a big area of interest for us. So, how do you create the experiment and how do you analyze your ideas and troubleshoot? We study sciatic nerves. Sciatic nerves are the longest nerves in the body. It's the legs from the foot all the way to the spine. And in animals, we there are a number of established models of nerve injury, like by application of ligatures, for example. Then this is tested very much like in humans for paw withdrawal. So the stimulation of the paw causes withdrawal. So if there is pain, then less pressure could be applied to elicit pain. And then the tissues are collected, and there are a variety of ways in which we can analyze. This is an example of histological analysis. Then we can isolate individual cells and analyze cells in culture. And then there are a lot of other ways, molecular and functional forms of analysis. I'll show you some examples. So once you've done all this research, literature search, and understood the scope of the problem, you want to focus and refine your question. The question that interested me, what is so different about nerve pain that we don't see in other tissue types of pain? Are there unique structures to the nerve? And my interest was with myelin. Myelin sheath is the insulating sheath around axon. So here you see the cross section of the sciatic nerve. And you see two types of axons. These ones, they're small axons. They're pain, called C fibers. They're slow conducting. And this in green, you see this is one cell supporting four individual axons. And on the right, you see much larger axons. And once they become more than 1 micrometer in diameter, this cell, the very similar cell, Schwann cell, start wrapping its plasma membrane around the axon, forming the sheath. So this myelin sheath is highly functional, and I thought it would be neat to show that we use these biological findings implemented in engineering. This is very similar, like an electrical cord, an insulating sheath that we developed for electrical cords. So what does myelin sheath do? Once the the stimuli, mechanical stimuli is accepted by the, say, the finger. Then myelin sheath prevents the electrical conduction. So the electrical signal jumps from one gap between myelin to another, and these gaps are called nodes of Ranvier. And this is called saltatory conduction. You can look it up. It's even on this YouTube link, very detailed demonstration of how it happens. But this is a process of speeding up conduction of the signal. So what happens is that myelin prevents these sodium channels, they're called, these molecules, these gated channels that are important for electrical signal. And what we observed is in normal nerve, in blue, this is the immunostaining for myelin, myelin basic protein. You see normal nerve, there is the myelin sheath, and in between the myelin sheath, there is the sodium channel that is necessary for pain, normal pain conduction. What happens after injury when the myelin sheath is gone below? You see a lot of sodium channels just spontaneously cluster in places they shouldn't, and that causes spontaneous pain. So this was one established way by which myelin was considered to be a part of this abnormal nerve injury neuropathic pain. And when I looked at this data and data like this, my question was, what happens to the myelin basic protein when it's degraded? It doesn't just go away. It creates fragments. So the hypothesis that I created was, can fragmented myelin be the cause of pain? So we went on to test this hypothesis. I'll show you just a few slides of how do you go about testing an idea like that. First we contacted our colleagues, Alex Strongen and Albert Rimaco there, who are experts in proteolysis of myelin basic protein. You can see this is a gel electrophoresis we're looking at. So basically proteins, they form these bands once you separate them on gel electrophoresis. I don't know if you can see my mouse moving. So what we see is that when we add the protease, this normal large protein gets degraded into 6 fragments. So we then submitted it to mass spectrometry, identified those fragments. So now we know there are these fragments, we call them 84104, 6884, and 2 and 12, that are formed as a result of proteolysis. Next question. Let's check if these peptides can cause pain. So we synthesize these proteins. There are companies that do that. Then we injected them into a normal nerve. And then we tested using the same von Frey testing I just presented you earlier. So what we see is that pain here, basically on this graph, you see low numbers means pain. What it means is that it takes only very low force for the pole withdrawal in animal to occur. And when there is no pain, it takes much longer. So much stronger stimulus, 15-gram force. So here we see that one peptide here, 6986, after a single injection causes at least 2 weeks of pain, a robust effect, as strong as the whole nerve injury. By contrast, 218 did not cause pain. So we see a very clearly specific effect due to some of the fragments, and very, very robust effect, as strong as the whole nerve injury by a single peptide. So this is part of people are part of my team, Zhenya Waching, who did this experiment. Finally, we ask a question. if this is the case, then by inhibiting this proteolysis and these matrix metalloproteinases or MMPs, these are proteases that degrade myelin basic protein. So if we inhibit them, if we block the activity by using this drug, this medicine, SB3CT, then this will go away. And true, we see that the effect of SB3CT in red, that pain goes away. So this is how you test a hypothesis. Now we've found a new target that is very, very effective in specifically neuropathic pain. And by blocking its proteolysis, we can prevent the development of neuropathic pain, which is very important because, as I mentioned, there are no effective medicine against this type of pain. So examples of youth research. It's a tricky question to answer because I don't really know what youth research is. Research is research. So the way that I see this question is let's see what the scope of the project is. What takes from the idea to a publication? This is basically a paper, complete, and now available for everybody as a conclusion. And then we can see where could youth, so to speak, participate. So first, the idea, as I mentioned, choose impactful idea. Choose idea that will change the field, change the society that would matter to you. Do research. PubMed is the standard of the field. AI is not, ChatGPT is not sufficient to provide rigorous assessment of what the status of the field is, or Google Scholar. So then we design the experiment, we do the experiment, we collect preliminary data, and we submit for funding. So funding can be submitted to the government, like NIH, VA, DoD is in the United States. Every government has this opportunity. There are individual foundations, there are universities that are going to look at your plan, at your hypothesis, at your data, and evaluate. So it usually there are people who are experts in the field. I'm sorry. Um, so this gets funded, and then we now need to proceed conducting the experiments. There is a committee approval for safety, animal or human research, and then we do the study. So each study consists of from 5 to 15, I counted several of our papers, different methods. So you put all these different methods to construct a story to have a conclusive data. So one project is approximately a year. And then we write it up, we create figures, and then we publish. So for the youth, what would be sort of your commitment? What would be your interest? Is it just a month you want to come in and participate and observe and maybe do medical search? Or are you there to stay for longer periods of time and contribute to data collection and design and so forth? So these are some of the methods that we use. I've mentioned 5 to 15 methods. We do surgery and then drug and gene delivery. We do functional analysis, then we can collect cells or tissues for analysis. Typically, youth will be not as involved in surgery, animal research, because it takes quite a bit of training and commitment. So it would be sort of in college as part of your sort of larger project. For example, we have at UCSD several such courses that could come into the lab and conduct this research. But what in my lab typically youth participates in is histology, for example. This is normal nerve histology. You can do it in plastic tissue, and Jenny here is the one who typically trains people in both doing the cutting of sections, staining the sections, doing imaging, and we will analyze it together. This is an example of a cross-section of a normal nerve and the injured nerves. So you see a lot of axons. You see V for the vessel on the left. This is how a normal nerve looks. A lot of myelinated, uniform, beautiful structures. And when the injury occurs, you can see on the right, there are immune cells infiltrating, like in this case, macrophages. We can use another method to show exactly the same thing. This is just staining macrophages, so using specific antibodies against proteins that are only found in macrophages. In this case, it's called CD68. So you see normal nerve, you don't see macrophages. It's a normal nerve, almost no macrophages. But with nerve injury, there's a lot of immune cells coming to help repair the nerve. And so this is what you see on the right. So this is very typically, this is done in paraffin tissues. We will do sectioning with students, and students can participate and learn how to do this. Or a little more involved, for example, is isolation of neurons. Here, Swati conducted sensory neuron cultures, and you do a lot of different ways of, you can add um, uh, in proteins or RNA of interest and see how these cells respond to that, and test all kinds of hypotheses. This will involve some training in sterile technique, and um, cells are like pets. They need to be fed every day. So for the duration of the experiment, usually a student would feed them every day and or collaborate with another student to take turns. But so this is basically youth research is contribution to a larger project to the degree of interest and commitment. And yeah, so most of our students participate in our outmost current projects. So yeah, this, if you want to sort of think back to the unsolved biomedical problem you like to solve, that I invited you to do in the beginning, think in this context. Think, what would it take to design an experiment and conduct it and do the research? Some of these things you can do without coming into a lab. So finally, how do you move from the examples or from the sort of youth research part, small parts of the projects, to creating your own program? And I can tell you a little bit about my path. So you can see sort of how does this career in a way unfolds. This is my first lab with Dan Santee at UC San Francisco. And there was a big lab, maybe 20 people. I'm only showing Dan here, where I learned how to mutate a gene. Basically, we get a gene and modify, do site-directed mutagenesis, modify this gene, and then we express it in the bacteria, gene expression in something like E. coli. Then the bacteria creates a protein. We purify this protein, and then we test for, in this case, it was the enzyme called thymidylate synthase, and we look for functional change in this enzyme. So the question with this, we would ask, what does this amino acid, what does this change, a mutation in one gene, can do to the function of the protein? My friend at the time was in the lab of Robert Stern, so I participated not directly, but I was attending their meetings, and then there I met my second mentor, Susan Hawkes, who studied extracellular matrix. And there I learned other techniques, new techniques. I learned how to culture cells and analyze them in culture. I learned electrophoresis, for example, here, Western blot. You can look up, all of these are available, of course, online to understand deeper the methodologies, but you allow the protein to separate, take a tissue or cell, homogenize, make it a solution, and then let it separate, let protein separate based on their molecular weight. And then you have specific antibodies here, LXR protein and GAPDH. These are just the names of proteins. And it's going to detect this protein very specifically. And you can analyze the changes in its levels and see and relate it to its function. But more importantly, in these labs, I learned about extracellular matrix and the fact that outside of the cell, there is this matrix of proteins that is so vibrant and so functional in changing the inside of the cell. That was very important for cancer biology and understanding how a normal cell becomes a cancerous cell at the time I studied that in the 90s. So, how did these experiences, my youth projects, so to speak, took me to developing my program and my lab? Finally, I came to lab of Robert Myers at UC San Diego for my postdoctoral work, and here I learned about nerve injury, about neuropathic pain, and behavioral tests and neuropathology. But what I did, I brought the ideas of the role of extracellular matrix instead of cancer biology here. I propose that it may play a role in neuropathic pain. And I was the first person to do that. And I'm still studying this. So basically, I brought the technologies and the concepts from my youth experiences into this world of neuropathic pain and started to test these ideas. So this is my lab. It changes because I do have, you may have seen in the introduction, I had 50 wonderful trainees. So many of these people come for a period of time. Postdoctoral fellows or graduate students can stay there for several years, and there are some. We have two Korean anesthesiologists here for come for a year of sabbatical. We have undergraduate students who may be just visiting us for a few months. So everybody has their own set of expertise, technical expertise. And then in our university and across the globe, we have scientists with, deep expertise, top in their field, and we can just contact them and ask them, tell them about our ideas, hope that they're interested, align our interest, and then start collaborating. So these are some of my collaborators over the years. And uh, with the project that I just talked about myelin, this project has been funded for many years, and these are my main collaborators, Alex Strongen and Tony Yaksh and Andrei Chernov. We have published 22 papers and created 2 patents and numerous presentations based on what I told you, but certainly we've showed many, many mechanisms in addition to those I was able to tell you today about. So as I mentioned, one of the reasons I absolutely love this work is because of its outreach. This is just the example of people who were reading that week our work, and you can see how international that is. And I know that some of you are also across the globe, and thank you for joining. And I think this is just so very important. We also have forums in among our colleagues, like any field. There are established forums where people who study, their students, there could be undergraduate students or the professors, everybody who has ideas or have questions, just go into these forums and we can discuss how best to conduct the experiments, how to troubleshoot the experiment. So this is a truly global community. And here in San Diego, we have a unique, one of the only two in the US, research hubs with several top institutions and biomedical companies sort of contribute to highly innovative work. And then we share. A lot of this work is shared in publications, but also what's called open databases available to anybody to just take and use for their own ideas. One point I wanted to make, I just gave you one example, but there are many programs that run at the same time in one lab. This is, for example, one other project I was very interested in about this more of a bioengineering, about the ability of metal, titanium, integrate with bone. This is very widely used in implantology. And we were very fortunate to work with PI Brainmark and his son, Ricard, in dental implants. These are the PI was the father of implantology. So we developed a rodent model, my mentor, and prior to my coming to this lab. And then we looked at the effect of extracellular matrix here on the right. These are some of my publications related to that, where we saw that, when metal integrates with the bone, there is new matrices formed, and specifically related to titanium and not other metals. So my final notes are just very general. What I think is very important in this line of research, of course, is knowledge and digging deep into the areas of interest. I think it's very important to study things that interest you and keep looking for things that you feel moved by and you think about. And wanted to say that, knowledge is everywhere, even today, for, showing up here, you might have learned some new terms. We talked about peripheral nerves, sciatic nerve specifically, myelin, different types of pain, neuropathic pain, Schwann cells. I'll talk about them in a minute, and different techniques may be new to you. So knowledge, people. I think this is just unfair to some of the people who I feel inspired by because I have to choose. But there are people who role models, Mary Bundy, that I don't even know her well. I didn't know her well, but her work really inspired me. Our conference communications inspired me. I have collaborators that are like Tony Yaksh, who have done just very, very important work in the field of pain. And it's a daily, really stimulation and inspiration to discuss ideas and to come up with new experiments and test them. And this is Jenny Dalkas. I was saying that inspiration doesn't come, it could be just qualities of people that inspire you. And Jenny is just wonderfully caring and fun-loving and determined and dedicated. And I know she makes my lab really sort of warm and welcoming, and I admire that about her and always inspired. And then here I've been very fortunate to meet these Nobel laureates, these and more, Gary Mullis and Francis Crick, and they're in categories of heroes. And I wanted to say not everybody has access to Nobel laureates, but even though they are here, they're in every institutions, in many institutions you'll find them. But there are heroes that accessible to us. Um, and I think it's important to be connected to your heroes. I wanted to bring three that we've touched upon today. Um, Schwann cell, I've mentioned, is the cell responsible for creating myelin sheath. Was first observed by Theodor Schwann in Germany, and he was also the first to suggest that it's not just plants that have these structures called cells, that animals do too. So he was very monumental in the field of cell biology. He also introduced the term metabolism and found the enzyme pepsin. Um, my second hero, Santiago Ramon Cajal, um, is called the father of modern neuroscience. On the right, this is the beautiful brain, is his book. I actually have it here in my office. There. Look, just beautiful illustrations. He was a physician, a scientist, and had the first access to microscopes. So he conceived of the idea that uh, the nervous system has specialized cells, and that were later called neurons. He also discovered a lot of important structures in the nervous system, like growth hormone and dendritic spines, and conceived the hypothesis of neuronal circuits, and have Cajal bands and peripheral nerve are very important functional structures. And finally, I mentioned Descartes, who also is a father. We have fathers of modern philosophy. You know him from his famous phrase, I think, therefore I am, a philosopher, a scientist, a mathematician. He was very interested in the idea of mind and body connection, and proposed to segregate them, and really was the first to um, dig into this body as the machine, conceive of this hypothesis, and describe many, many different um, biologically and medically revolutionary ideas, including pain. So my heroes. Um, be inspired as a student, get engaged. Labs are available. Don't hesitate to approach labs, approach your professors, PIs. As a high school student, as a undergraduate, as graduate, I will have three high school students joining me for our lab for the summer. So just approach um, professors whose work interests you, read about it online, and live fulfilling life is my advice as part of the biomedical research discovery. Um, I Same for professors. I wanted to know that as professors, we have three legs of the stool, as they say. We do research, we teach, and we do service. Um, and so I participate in these also thoughtfully and do things that I only find meaningful. This is uh, something that I wanted to say about academia and university, where you have an opportunity to participate um, in so many different departments and interact with different colleagues, not just in biomedical research. This is an example of my participation in Human Condition Art magazine for many years. I contribute my photography. I was on the thesis defense for screenwriters, which is just a different world, and they love it so much. So strive for excellence in your field. Um, Take risks. I think failures are very important. The sooner you know how it feels, the better adapted you are. And sometimes it will allow you to choose the path that you prefer, you know. And something I advocate. I don't really, it's a very fashionable work-life balance term. I don't understand it because to me, this is sort of when you love what you do, the work friends are friends, and we have a lot of fun, and we do a lot of travel, presenting our work. And at this point, I will invite questions. Um, I can stop sharing.
I want to say thank you so much, Veronica. It was incredible journey into the world of medicine, biology, and discovery. We appreciate your time, your energy, your experience, of course. And right now, guys, if you have questions, you can ask Veronica. Якщо для вас дуже тяжко, наприклад, говорити англійською, Veronica can speak Russian. So we can ask English, Russian. If you have questions, please ask right now. Питайте зараз.
I need to stop sharing too. Stop sharing.
Якщо є питання, питайте. Нема питань? Це було дуже чудово.
Я вибачаюсь, Оксана Мущенко. Я прослухала, яким чином приєднатися. Я там чула, що є програми для high school students. А як приєднатися до цих програм? Я якось вибачаюсь. Ми просто в дорозі зараз. Я прослухала. Чи можна повторити або дати інформацію, де більше можна ознайомитися з цим? Я думаю, що наверняка десь воно на сайтах є офіційних.
I understood part of it.
Будь ласка, якщо можете запитати на русском языке.
Какие-то программы, если программа на...
Да, если есть программы, где можно присоединиться к программам именно для студентов high school, и где их можно найти? Где можно найти эту информацию?
И это в зависимости от того, где находится, это в Украине или здесь? Где, о чем речь?
Сан-Диего, я думаю.
Сан-Диего. Есть VCSD-программы и есть программы в самих high schools. То есть это я буду рада ответить на кому-то, и вы можете меня сконтактировать, если в зависимости от вашей школы. Например, в Лахоя Хай есть Biomed Program. У нас обычная молодежь оттуда.
We live in Encinitas. It's San Diego High School Academy.
Okay.
He studied here. He also it's about my son. He also participates in math circle in the university. Yeah, correct.
Okay.
Yeah, and but I know math circle have like official website, and we take information from there. What time they can apply, what time they can enjoy, and etc. Yeah.
Lenочka, if you have my contact, just please leave it and they can help with directing
Добре,
these.
просто напишіть мені, я дам тоді вам якісь посилання корисні, добре?
Окей, дякую.
Дякую за питання. Спасибо за вопрос. Thank you for your question. If you have some question, ask right now. And I just want to let you know, guys, that форма зворотнього зв'язку ви можете знайти у чаті. Будь ласка, натисніть на це посилання і надайте фідбек. Це дуже важливо для нас. It's very important for us to give a feedback from you.
I had a question.
Yes.
I wanted to get involved in research over summer, and so I live in San Diego, and I'm transferring to UCSD in the fall. And I
Uh,
was wondering, like, what's the best way to go about asking a professor to get involved in research, like by email or in person, or like how should I go about that?
I would suggest looking into the area of your interest and finding professor or maybe one more than one, and directly emailing them a letter of intent, sort of, or it could be a short email. Hopefully, with I think it always helps if you show your interest specifically and indicate that why this research interests you. And yeah, I think it's just a matter of contacting professors and looking at the opportunities.
Okay, thank you.
Yeah, my pleasure.
Thank you for your question.
May I ask, is there any specific interest of research that you are considering?
I honestly am interested in your research because I like the neurobiology kind of aspect of it because I took anatomy this past fall, this past spring,
Mhm,
and I really liked the neurology part of it.
mhm.
So hopefully something in that.
Okay. Well, I'm happy to direct you or to answer your question or to meet you. So don't hesitate to ask me, but I also can help you uh, with contacting other people if you don't succeed.
Okay, what's the best way to contact you then? Because I'm
Uh,
not sure if there's an email sent or
yeah, I can type it. There you go. It's in the chat.
thank you.
Yeah, my pleasure.
Thank you for your question too. So let's check our chat, and you can find the form of feedback because it's very important for us. And you can find the Veronica email if someone interested about that. Okay, so if we don't have more questions, we can finish our webinar today.